Dear viewers, greetings. In this video, we are going to see the various cell inclusions in bacteria. Within the cytoplasm of bacteria, several kinds of reverse deposit are present and they are known as cell inclusions or inclusion bodies. The bacterial cell may accumulate certain nutrients when they are plentiful and use them when the environment is deficient. They will collect the nutrients and save plentiful of nutrients within its cell. If in case any deficient will occur, means it will utilize that particular nutrient. That kind of nutrient is referred as the cell inclusions or inclusion bodies or inclusions. Some inclusions are common to wide variety of bacteria, but some other are limited to small number of species. Therefore, that particular inclusions are served as a basis for identification. The very well-known example for this is metaprobatic granules. Regarding the types of cell inclusions, there are 5 types of cell inclusions are available in bacteria. The first one is the metachromatic granules. Second one is polysaccharide granules. Third one is lipid inclusions. Fourth one is the sulfur granules. And the final and fifth one is the carboxyzones. The first type of cell inclusion is the metachromatic granules. It is a large inclusions that take their name from the fact called as metachromasia. That means the certain blue stains such as methylene blue will stains the particular strain and it reflects the strain as red color. This kind of change is called as the metachromasia. And the metachromatic granules are made up of polymetaphosphates and also called as volutins or volutin granules or Babes Ernest granules or polar bodies, and they are represented as a reserve of inorganic phosphate, particularly the polyphosphates, that can be used in the synthesis of ATP. They are formed by the cells that can grow in phosphate-rich environment, and the metachromatic granules are present in bacteria like Corynebacter diphtheriae, Spirillum species, Rhizobium species, and some Bacillus species, algae, fungi, and protozoa. The second type of cell inclusion is the polysaccharide granules. They are also called as polyglucan granules or hydrophilic granules, that is water-loving granules. It consists of glycogen and starch. In the presence of iodine, the glycogen granules appear in red color and the starch granules appears in blue color. They are deposited by the bacteria themselves in the cells when simple sugars like glucose, sucrose or fructose are present for polysaccharide synthesis or glucan synthesis. And this type of polysaccharide granules are present in Clostridium species and some coliform bacteria. And these granules act as an important source for the carbohydrate metabolism in the bacteria. The third type of cell inclusion is lipid inclusions, which are also called as pseudophilic granules or poly-beta hydroxy butyrate in short form PHP. They are revealed by staining the cells with fat-soluble dyes like pseudon dyes, so they are also called as pseudophilic granules. They are formed during the lipid synthesis, acetate or butyrate metabolism, nitrogen deficiency conditions, or denitrification process. And this type of lipid inclusions appear in various species of Mycobacterium, Bacillus species, Rhizobium species, Acetobacter species, Spirillum species, Alkaligen species, and other soil bacteria. The fourth type of cell inclusion is the sulfur granules, which are also called as the sulfur globules. And these sulfur granules are the cytoplasmic globules of the element sulfur, and they are present in the sulfur bacteria, which are widely involved in the sulfur cycle that belong to the genus Thiobacillus thioxidans, Chromatium species, and Chlorobium species. And these sulfur bacteria derive energy by oxidizing the sulfur and sulfur containing compounds, and the sulfur bacteria may deposit sulfur granules in the cell where they serve as an energy reserve. The fifth and final type of cell inclusion is carboxyzones. They are the photosynthetic bacterial cell inclusions that contain the enzyme ribulose 1,5-diphosphate carboxylase. This enzyme is an important enzyme for the Calvin cycle or carbon dioxide fixation reaction. The photosynthetic bacteria use carbon dioxide as their sole source of carbon and require this enzyme for the CO2 fixation or carbon dioxide fixation. Some of the carboxysome containing bacteria or nitrifying bacteria, cyanobacteria and thiobacilli. These are the carboxysome containing bacteria. Thank you for listening and support. Thank you.